Good morning. I didn't put my name down because I'm not sure who I am. Really. So I'm Mayank Mehta. I live in multiple departments, and my goal will be to convey to you as one of the major challenges that we have, which is how does the AI actually work? We don't know it. It's too complicated. And that problem we have faced in the brain already. We're using it right now. It's another example. We're using this hardware without knowing what the hell it does. So I'll convey to you some things that we can do. In fact, something that we can do at the end of my 3 lectures, if you manage to survive that long. I guarantee you some of you will fail. And that'll be a testimony to how interesting this got. At the end, I will try to show that actually you can calculate some things rather intuitively that works in behaving brain at microscopic level. So let's get started with those over promises. So that's where we want to go. That was my life before I started to do this. In physics, I did PhD in quantum field theory, and the world is simple. Well, it looks a little complicated, but it's pretty simple. You have a handful of particles, you have a handful of equations, and everything is beautiful. As we learned in mathematics, we want to go for beauty, and it's simple, beautiful, and all that. Everything works. Well, there are a few problems like dark matter, but who cares? All right. Little details, all right. But if you really dig into that, this is equation number 53 from my PhD. Not very pretty. Not very pretty at all. So the answer is not 42, it's d minus 3 times 2 pi. That's about it. So that's what we want to do. That's what we want to do here, the basic concepts that are there, and then we can put together, and at least we can convince ourselves that we can calculate things, we can hold it in our brain and turn it around, and when we are showering or taking a walk or watching a boring movie, we can do math about the brain. So that's what we want to do. And if we trace the history down, all this happened because before that there was Newton's laws. In variety of things, Newton, I mean total anomaly, thermodynamics, calculus, mechanics, all that, but before that there were Kepler's laws. And I believe that's where a major break happened in science. People knew things were going around, and there were a bunch of anomalous particles like planets, and there were some trajectories, look more or less spherical or circular or whatever, but he came up with the exact law. Insanely, 2/3 power law, and it works across all the damn thing. What the hell is that all about? Why the hell are these things so simple? 2/3. What's up with that? And why, I mean, our brain could do 2/3, animals can do that. Speaking of intelligence, Konrad Lorenz, a famous behaviorist, did the simple probabilistic experiments. He took him and his friend, they stayed by the lake, and they started throwing breadcrumbs in the water. Ducks came in. They started to throw breadcrumbs at the same rate, more or less equal number of ducks on both sides. One of them increased the rate at which they were throwing the breadcrumbs by a factor of 2, became 75-25. So they did that quite a bit. So somehow, ducks just screaming and squabbling and all that stuff, they could figure out probabilities and the nasty duck and all that business. So there's something going on there where we are able to do something very simple at a level of population, compute probabilities. So, and before that happened, there was Galileo. And I'm going to try to say, hopefully this is not being recorded. Oops, it's being recorded. That's where we are in the brain science. We are at the Kepler's level, not really there. We haven't had that 2/3 level of breakthrough yet. And we have already gone down to all this level because we can do math. And my goal is to convince you that we need to slow down, get the equivalent of Kepler's laws, and then do this. So I'm going to be holding, for those who are interested, a whole bunch of tutorials. I'll be giving you exercise. I teach a course on neuro-AI every year. If you want, you can come to UCLA and attend, or remotely. And I run an interest group on neuro-AI, where our goal is to start here. And if one of you becomes Kepler, my life's mission will be accomplished, because I can't be, I'm told. All right, stare at this. What's here? Don't give the answer. If you see what's here, raise your hand. 1, 2, 3, 4, 5. Not everybody yet. Yet? Yes, thank you. I'm counting the distribution. Anybody else sees this yet? Anybody else sees this yet? Yep, nice distribution. Excellent. Thank you. Now, thanks for some of you who could not see this. Let me enlighten you. The only time, that's a Dalmatian dog right there. That's his head. That's his ear. Those are his legs. That's a hind leg. That's maybe the tail. That's the shadow of a tree. And there is some other crap on the floor, right? Very simple exercise, black and white. There is no gray here. And your brain perceived it. And I changed your brain permanently. Trust me, I did. Because if you were to see this any time later in your life, you will recognize this way faster than you did just now. So, yes, please.
Does it say anything about the way some people's brain process?
It says and it doesn't say. We are not there yet. We are not there. I mean, that's the way it is, right? That's a nice challenge. I don't say it in a negative way. Is that now the technology has become available that we can ask these questions. And I will get into that a little bit. Yes, please.
I don't believe everyone saw a Dalmatian. I think they just raised their hands.
Now that's another interesting point that you raise. It's very hard for us to put ourselves in other person's mind, right? If I saw it in 2 seconds, I don't believe other person didn't see it. Maybe they didn't clean their glasses. Maybe they were looking at their phone. And I'm going to tell you that actually the same problem is going on in your brain itself. Forget across the people. That social business and so on is actually going on in your brain. We'll get to that. Any other hands that were there that I didn't? Yes, please.
Oh, just to respond to Donnie. So actually there has been a lot of studies on optical illusions in general, maybe not necessarily with particular images. And people with schizophrenia don't perceive certain illusions, for instance. So there is a correlation also in, not this one, but there is certainly some correlation that has been shown. And if you're interested in that, there are people that are actively studying that. And it's just cheap to do because it's behavioral studies.
There's a lot going on, as you will see. So let's get into that. Yes, please. Sorry.
Yeah, the funny thing is I see something like a collie pointing the other way.
Yeah, that's right. That's right.
The thing is sort of like another dog.
That's right. You see a lot of things, right? And this gets into, let me not get into what this gets into. This is still an introduction. This is great. So please keep these questions coming. But all right, let me just turn this blank, change of program. So what happened just now? What happened? I was going to come to that, but why don't we get into that already since you raised the question? There was a low-dimensional image. There was a bunch of pixels, very handful of pixels, couple hundred pixels, black and white, zeros and ones. They fell on your retina, and then there was an explosion of dimensionality. You have about 100 billion neurons, give or take. And out of that came one number. What is it? So now, maybe 2 numbers, maybe 3 numbers, Dalmatian, count 1,000 possible names. Nothing compared to 100 billion. So what's going on when the brain is operating is that we live in a world, as far as we can tell, and I'm going to shake those dimensionality a little bit, we live in a world which is pretty low dimensional. You look at most of the images, million pixels, nothing. It's nothing. Our retina itself has a very small number of pixels. And we don't even see all the pixels, even though in principle we can. And from there is an explosion. From that CCD camera, more or less, already, not going that way, but let's just run with it, it gets thrown into a giant amount of explosion. And then the number of behaviors we come up with, couple thousand words, couple hundreds of hand movements, couple dozen foot movement, and that's behavior. So there is this fundamental problem of massive explosion of dimensionality and then massive collapse of dimensionality. Pretty tough problem. And many people are working on it as well. So that's what's going on, and that's why, if I were to pontificate a little bit, AI seems to be reasonable when the answers are not very precise. Because the dimensionality is rather fuzzy, but if you collapse it too much, it fails. But let me not get into that. So let's get started with this. So why? How are we able to do this? So let's put some numbers to it rather than just pictures. So what's so special about the brain? It's about that big, 2 fists, heart 1 fist, brain 2 fist. Pretty easy to understand. Most people would say 100 billion neurons. But well, if each neuron is more or less 0 and 1, which is what we think in most of the time, people have made computers with more than 10 raised to 11 units, and they are nowhere close to us. So it's not the number of neurons. In fact, my foot has easily 10 raised to 11 cells, nowhere as interesting as the brain. So it's not the number. What else is going on? Maybe the brain is super fast. Not true, actually. The clock speed in your laptop or whatever is gigahertz or more. Your brain's clock speed is, as far as we know, kilohertz at best. So massively slow, like insanely slow. Then the energy it consumes, 20 watts. This is 20 kilowatts, at least. Insanely efficient. And then things I can't put into pictures, we get exhausted. That's why people want to make AI. They want to get rid of us, so that we are not all these pesky little things. Oh, I'm tired. I need a break. Nope, keep working. So, right? That's what I tell my grad students, work. So, no. Maybe I do. No. So we get sleepy. And now we have intuition. Yes, I got tired. I'm sleeping. But the amount of energy that your brain is consuming is only 10% less than when you're awake.
What do you say 20 kilowatts? It's way too much. No, I mean, a single chip is on the order of like 20 billion transistors, right? Now.
If I look at one of these, it depends. It can be less depending on which kind.
Small as the chip on my phone. That's not 20 kilowatts. It's like
not on your phone, but when I go to some giant number of transistors.
Free chip. Like in the
That's right. So that's right. So if you want to if you have to have a chip which is programmable, which does some kind of computation, then the power demand goes up. If you simply have a simple chip which does something pre-programmed stuff, it is less. Absolutely. But if I want to do something which is complicated calculation, it goes up massively. That's why people need giant cooling towers and so on to operate many of these things. You don't seem convinced, so we can come back to it. We can say maybe, may not be. Totally fine. So, yes, please.
Just to clarify, that 10 to 11, you're not referring to like this phone is, from your count, is not 10 to 11. The 10 to 11 is...
It's some giant machine. That's right, some giant machines. That's right. Yeah.
You say 10 to the third hertz. The microtubules actually are 500, are known to be 500 megahertz. So that's kind of dangerous.
That's right. So let's come into where this kilohertz come from. I'm going to show you why people think it's kilohertz. And there are people and Penrose and others who talk about microtubules and consciousness. I think
but there's papers that have published it.
I know. But does it influence neural function? That I'm not sure. And the answer is no. You can come to that. Yeah, I know. Yes, Penrose is great, and he has written these papers about quantum consciousness and so on, and I love quantum mechanics, but has no room, in my opinion. But let me not get into that. So let me just pretend that there is something going on, and for the purpose of this talk, I will stick with kilohertz. How about that? For the purpose of this talk, I'm going to be talking about phenomena that occur at kilohertz, and I'm not aware of anything that in vivo anybody has measured which is going much faster than kilohertz. I'm not aware of it. I could be ignorant.
I can send you papers.
Absolutely. And I have read some of those papers, and they are in my trash bin. But sorry. I know I have read recent papers. Sorry, but please bear with me. If it's okay, let me just go with kilohertz calculation, and then maybe we can go to something more interesting. And I'm interested in microtubules and quantum mechanics. I did a PhD in that, so I'd love to see it. But after 25 years of looking for quantum mechanics in the brain, it's not there. Sorry. Okay. So then, when we are sleeping, your brain is still going on. So you can say, well, all right, when you're sleeping, your heart is pumping, your gut is doing its stuff, breathing has to happen, it's all happening in the brain. But turns out your eyes, the visual cortex, your eyes are closed, visual cortex is going gangbusters, more or less 20% less than when you're awake. So what's going on there? We'll get into that a little bit, but at the end, we conclude that the brain, with my ideas, is slow, sloppy, sleepy, but massively efficient. And that seems to be an evolutionary purpose, because if I'm an animal, and if animals are moving around, when they move, they spend energy, I better know that when I'm going to expend energy, the net energy that I get is more than the energy I spend. Otherwise, I'm dead, right? Then I'm out of the evolution and nothing happens. So we need to conserve energy, and I think that if you want to understand how the brain works, this is a cardinal principle we want to start with. It wants to conserve energy, wants to maximize energy. That's why I eat ice cream and cookies and fat, and we are programmed to do that. So how do these things work? So let me actually start with some simple idea. Since I'm told not to go into the modeling part in mathematics, somebody else will cover, so I'll try to not go too much into that, just talk about the basic science. So the way the neurons operate is very simple. There is a certain amount of current. The neuron, as a result, generates some voltage, and if the current exceeds some amount, if there is a small amount of current, the neuron doesn't care anything. Exceeds some amount, it shoots up, and then it kind of saturates. So that's your ReLU operation, zeros and ones, and all that stuff. But actually, that's not the whole story. And the brain starts to deviate from this right at the most fundamental level. So let me see if I can figure something out, because this slide should be here and didn't go through. So how many of you have actually heard an actual neuron in the brain? A few of you have. So this is the sound of an actual neuron while there was some stimulus that was going on the screen. All right, I'm going to repeat this. Have a seat, and I'll play this again. It's rather faint. So what's going on is that on the screen, somebody is doing this. There's an animal. Please have a seat. There's an animal that's sitting somewhere, and somebody just moving a bar of light back and forth. That's it. That's all that's going on, nothing fancy. The animal is anesthetized, so no pesky thoughts and intelligence and all that stuff. Just bar of light going around. So I'm going to play this again. That's what the neurons do, so let's see if I can go there. All right, it's too faint. All right, I managed to capture a little bit of it. So this is the subthreshold voltage of the neuron. Some current is coming in from senses somewhere. The voltage is fluctuating away all over the place. And then, every now and then, when the voltage reaches this threshold, it generates this nonlinear event that I'll not be able to capture, called a spike. So let me see if I can again capture it this time. There you go. So as soon as the voltage reaches this value, it shoots up massively. These are like, that's why people say brain is digital. This is 1. And all that is kind of 0. So if the voltage is bobbing around here, you will never get this. But if it crosses and these are specific numbers, brain more or less sits around minus 65 millivolt. That's not special to the brain. Most of the cells sit at minus 65 volts. News to you, maybe. The cells are not sitting at 0 millivolts. There are lots of complicated processes that are keeping that at minus 65. Bacteria cell walls are at minus 65. So it's, if the one were to describe the life with one number, it's minus 65, not 42. And it sits more or less there, and from there it goes to minus 55, and boom, it says, okay, I had enough. It generates this massive thing, and even though the amount of current is still strong enough to make it active, it goes to minus all the way to 100 millivolts. So each of this event is 100 millivolt in amplitude. This fluctuation is of the order of 10 millivolt. This giant thing called an action potential or a spike is 100 millivolt. So 10:1. And it lasts about a millisecond. And when this spike occurs, that's when the neuron dumps its neurotransmitter onto other neuron, and other neuron is able to listen to what the neuron is saying. When all this is happening, Nobody knows what the neuron is doing according to our current understanding. So neurons are connected by lots of synapses, as you all know. These synapses are sitting there silent, more or less doing nothing. And as soon as a neuron crosses this threshold for 1 millisecond only, and the amount of 0.1 volt, that's quite a lot, then it dumps neurotransmitter into everybody is connected, and they all know what the neuron is saying. If the neuron said yes, and that's the only answer. Otherwise, neuron says no, 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 yes. So all these are no's. This is a yes. That's when the network lights up. And on an average, the neurons generate about 1.5 spikes per second. That's the average activity level of neurons. 1.5 hertz is the magic number. So that's the other. It's a broad distribution. The activity levels of neurons is a log normal distribution. But on an average, they sit around at 1.5 hertz. So now that minimizes energy consumption quite a bit. Because this thing doesn't take much energy, but when it fires a spike, sends neurotransmitter, all kinds of chemicals have to be synthesized, some membranes have to be reorganized, those membranes have to be repackaged, consumes a ton of energy when the spike is generated as an after effect of spike and spike itself. So brain has found a way to operate at a level of 1.5 hertz, lowered the energy cost massively. But I cannot go into the math of this, but that poses serious computational challenge. And that can be understood very simply. We want to be Keplers, so let's think about it. So I'm one neuron. I'm listening to you. You are one neuron. I'm saying, hey, what's going on out there? What's going on out there? And you don't say anything, and once in 1.5 seconds, you say something. But my perceptual time scale is, blink of an eye, maybe faster. So how does this network give you stability and all kinds of things when this neurons are genetic spikes at the level of 1.5 spikes per second is an interesting, solid challenge that people are still working on. There are interesting ideas, but we can get into that some other time. So as a result of it, one sec, let me just finish. I want to put down Mayank Mehta's first rule. We can introspect about the mind, but we cannot introspect about the brain. This is a common mistake many scientists do. That, today I saw a wolf that looked like an elephant, so that's what happened in my brain. No, don't do that. We cannot introspect about the brain, we can introspect about the mind. Mind is what we perceive. If you ever felt a spike in your brain, go see a doctor. You cannot perceive a spike. So the brain has hidden its hardware from us completely. No chance. You can't even perceive but on the other hand, if I were to go and stimulate one neuron by hand, 1 out of 1 billion, I can change your perception. But I cannot make you perceive the spike. So it's carefully hidden. The spikes are happening in your eyes, you don't see things blinking. Spikes are occurring in your ear, you don't see pop, pop, pop going on there. They're giant ones, they're 0.1 volt. So exercise number 1, if there are 100 billion neurons in the brain, they are firing at 1.5 hertz, and each spike lasts 1 millisecond, and it's 100 millivolt in amplitude. How much is the electrical potential maximum possible in your brain? All right? For those of you who are students, try to run these numbers. That's the only way we learn. If you just think about it, nothing happens. You get to calculate. It's a very simple thing. Put these numbers down and see the total real potential of a brain. You had a question, sir.
So I thought you said, but I wasn't sure. So when a neuron fires, all the synapses
Yes,
fire in the same
almost. Well, all the synapses, the strict sentence is that the synapses of a neuron have a chance to activate only when the neuron spikes. When the neuron doesn't spike, more or less it doesn't. There's some spontaneous kind of noise release which you are going to exclude. But the second part, why is it that I don't say when the neuron spikes, all the synapses are activated? It turns out synapses are highly noisy. They have an activation probability of 0.2, more or less. So when the action potential arises, and it depends on which synapse, maybe somewhere it's 0.8 release probability, somewhere 0.2, but in that broad range. So synapses will not necessarily generate whatever current into the other neuron, but that's when it has the largest probability of generating current in the other neuron.
So, I mean, if you're modeling it, It's a big difference between saying it's the same activation energy for all synapses.
That's right.
Saying it's different.
That's right. That's right. So we have
You're
to.
saying it's different.
Yes, absolutely. And there's lots of research, books. There's a whole book by Hille called Synapse. With those who are interested, 20 years old book, but still going strong. There's a very fancy machinery in the synapse itself. We'll look into that a little bit. All right? Yes, please.
What were you just saying right now is quite stochastic.
Yes, very stochastic.
What is?
Everything about synapse is stochastic. It's been quantified in terms of three things. A, whether the synapse, well, let me throw a little bit of jargon at you. For those of you who find too much, don't listen, just cover your ears. Synapse doesn't just dump out chemicals. Synapse packages into little balls called vesicles. How many vesicles? Turns out for most neurons, there are only about 100 vesicles available. That's a very small number. Out of those 100 vesicles, the number of vesicles that can be dumped out, called the readily releasable pool of synapses, that are only 10 or 15. So now what happens is that when the impulse comes in, a voltage that leads to some calcium influx, complicated stuff, and the synapse vesicle gets dumped out. Now that vesicle, the neuron either has a probability, whether a release vesicle or not, or that's n, or the probability of release. 2, how many vesicles do I release? Because it's quantized, 1, 2, 3, 4. The number of transmitters are much bigger within a vesicle than number of vesicles. And once some amount of vesicles are released, now it takes a neuron like ages, like 100 milliseconds to replenish it, to bring it back to be ready to be fired. Like the old guns, people used to have you fire, and now somebody has to go and stuff it again to fire. That's what happens in the brain. So there's a strong history dependent in a synapse. Massive, on the scale of 100 milliseconds, but more. And that comes to predictive coding, which hopefully I'll come to eventually.
I guess the history dependence doesn't necessarily mean random. Like
that's right.
Tony Bell, for example, has an interesting take on this, where he's like,
that's right.
Oh, extreme.
So these debates go on endlessly, but nobody has really shown that here is one synapse, and I can tell you exactly when it's going to release vesicle and how many. Nobody has done that. So we don't know if it's necessarily stochastic or history dependence in a couple of calcium levels and so on can necessarily explain, but there are clearly many predictable factors as well.
And one other detail I'd be interested in, the vesicles have quite uniform amount of neurotransmitters?
No, different amounts. But that's much smaller than just the total amount. And there's also stuff going on the other side, which we are going to get into. Yes, please.
Well, this is kind of a silly question, but I remember hearing a long time ago when Beethoven would compose quite a bit, he'd put like cold towels on his head because he'd feel like he was overheating. His head was overheating. And I'm wondering, like, so you can't hear individual spikes, but I mean, is there enough sort of energy release where you, like, if you
there are
really sort of feel your...
There are lots of things. So now I'm going to venture into territory where you better stop recording because I'll get to jail if you heard this answer. So the amount of current that's going on is pretty small. So as a result, you can take a 9-volt battery, that's it, and take 2 wires, pass current in your head. This is called transcranial direct current stimulation. You can Google it. I didn't say this, but there are several people who will swear by it that if you pass more current in the frontal cortex, then it's better for them to focus, so they get some juice, and if you put here, then it's better to do meditation and better to be relaxed. So there's a whole thing there. That's right. Now, more fundamentally, if you look at how neurons generate spikes, it's described by a Nobel Prize-winning research called Hodgkin-Huxley equations. Hodgkin-Huxley equations have a temperature dependence of T to the power 4, massively temperature dependent. So there is a lot going on there, which we can't get into. Yes, please.
Yeah, where exactly are you putting the kind of voltmeter here on the cell? Is there a simple picture you could draw to show that?
Thank you again. I skirted all that. It depends on with respect to what. Now, the standard way it is that in this experiment at least, they put an electrode through complicated means that got a Nobel Prize in 1993. My longtime collaborator, Bert Sackman, developed it. So one electrode kind of goes inside the neuron. Now, getting inside the neuron is not tricky because neuron is only 10 micrometer cell body. So getting a thing into it is tricky. The brain is thumping around, so the neuron is dead. But anyway, if you do all this, the other electrode is somewhere at the back of the skull, roughly. That's roughly the ground. But that gets very tricky. It's not, every neuron is not really seeing the ground. Every neuron is seeing its neighboring neurons. So we have to start thinking of a flow of electrical voltages between neurons. Very interesting. I'll maybe come to that, but at this rate, I'll not come to that at all. That's totally cool. This is nice to have a conversation, yeah.
Sorry, maybe this just needs to be brushed aside, but so if you were shrunk, so in a hypothetical imaginary world, you were able to go in and literally place some magic voltmeter at exactly the places you would want, hypothetically, ignoring all the practicalities, where would those two ends of the voltmeter go?
What we do, I have done those experiments, so what we do is that, you put some saline solution, brain has a bunch of salts, several kinds, sodium chloride, potassium chloride, calcium chloride, that's mostly sodium chloride, that's why we need salt. So you put, there's a cranium here, so you put some salt right above the salt water above the brain, you put ground electrode right there. That's my favorite ground. When I measure signal in the brain, I put my ground electrode right on top of the neurons, right on top, which is within 0.5 millimeter, so now all the floating voltages are cancelled. And there's no brain matter above the skull, so I can calculate this. That's what we do. And I didn't invent it, that's what many people do. Those who don't do, I don't like them, because then they're putting all kinds of weird voltages in the brain. Sorry, you had a question.
I have a naive question, maybe. It's about, isn't there some kind of pruning process going on that would allow a person to find it optimal synapses to activate?
Don't know. Music to my ears. I would like to believe it, but we don't know. The fact is that we are born with all the neurons that we have, more or less, except two parts of the brain where new neurons are generated all the time. One is in your nose, right here, olfactory bulb, and the other is a deep part of the brain. We'll spend some time on it, which is the depth of the deep network in some sense called hippocampus dentate gyrus. Those are two places where new neurons are being born constantly at the rate of about 1 per minute. So already you got 35 neurons out of 100 billion, but hey, who is counting? Right there, I lost 1,000. I'm still fine. Now that's another thing about stability. But then the cool thing is that for the first 3 years of a life of a baby, there is a massive amount of synaptic explosion. Number of synapses are just growing. So neurons, the babies have the same, but the brain starts to grow because these processes called dendrites and axons, they grow, and synapses. Each neuron has 40,000 or 10,000 synapses, they are growing for the first 4 years, roughly. And then there's a period of synaptic pruning once again. The brain starts to throw away whatever irrelevant synapses, and then this process continues at a lower pace throughout life. That's what roughly happens. Anything else? All right. So that's what happens, and as a result, what has happened in the field of AI is that we have gone directly. We have taken one weird hardware, which has ReLU but doesn't have spikes. Nothing wrong with it, but these are major differences, and my goal is to tell you what is similar and what is different between the brain and machines. Machines don't have these zeros and ones, these kinds. They just have these voltages, sigmoid function right there. And then we are imposing the intelligence directly at the level of this. So we want the hardware itself to do error correction and so on, whereas in the brain, nobody's teaching anybody what that is. So I'm going to spend a little bit of time telling you how does the brain actually learn rather than me as a whole organism learns, okay? So with that, let me see where I can go, and I need to keep track of time. So let me tell you a little bit about this synapses, since there were questions about it. Now, here is something very funny. So let me turn this into blank screen and so on. So brain has neurons that connect with each other, not so special. There are cells in your body that are in contact with everybody else. We are jam-packed, we are solid objects. So what happens is that every cell in your body, if you assume close packing or the other, is in contact with about 12 or so other cells. And then unless it's blood particles or something, these cells are more or less the same. That's number one. So every cell in your body is in contact with roughly a dozen other cells, and its neighbors are fixed. That much is known, but now comes another fact. If there is a cell here, and that kind of just pushes the other cell is going to push right back. the connections or the interactions between cells are more or less symmetric. What I do to you, you will do back to me, more or less. Now, what has happened is, what's special about the brain, it's not the number of neurons, it's not the number of cells, but brain has gone out of the way, and this has happened much later in evolution, to create the synapses which are not symmetric objects. There are asymmetric objects. And I'm not aware of too many other places in the body where the evolution has gone out of the way to violate this fundamental fact, the Newton's first law of motion, what A does to B, B does back to A. That is exquisitely violated. So what happens here? So first of all, here is the neuron. That's a cell body, 20 micrometers. That's another magic number. Everything is cell sizes on an average, 20 micrometer. Bacterium, 20 micrometer. Virus is much smaller. But then the neuron has these gigantic processes. They are of two types. One are called dendrites. That's where the inputs arrive from other synapses. And other is the output called an axon. And the axon, as all you know, have this fatty tissue called myelin, which makes sure that energy is minimized. The action potential that's generated here travels all the way. And then these things make a synapse onto another neuron, or lots of neurons, about 10,000, give or take. Now what happens is that it's only the output at the end of the axon that dumps neurotransmitter. And it's on the other side that there is a receiver called neurotransmitter receiver. And the circuit necessarily goes only one way. So many of you have used neural networks. I'm sure many of you learned the RNN or recurrent neural networks or Hopfield networks or Ising model. Every physical theory that we have made in condensed matter physics is about a bunch of interacting particles which 100% respect Newton's first law of motion. That's why we can make Hamiltonians, we can make positive definite kernels, we can do regularization and so on. Brain has said, nope, I don't like it. I don't want symmetric connections. So the first thing that I will say for people like me, I used to be one of those who made Hopfield model and so on, say that's a serious problem for the brain because it's gone out of the way to violate that. The neuron itself is a polar object. All the dendrites are where the inputs arrive. All the outputs only go out through axon. In the early during evolution, like in the fly brain in the evolution, the input and output are not kept separate. They're mixed in. So through evolution, the brain figured out that input and output need to be kept separate. And then on top of it, not only is the neuron a polar object, but every connection here is also going more or less only in one direction. And again, in biology, nothing is 100%. There are clearly counterexamples and so on, but this is like 95% at least. Yes, please.
Is there any loop in which this neuron influences this one, and then it...
That's right. It can happen, and most of our theories are based on that. But that starts to get into trouble with all the synaptic weirdness that we discussed. So those loops, we make theories about it, but don't really work. And I'll eventually maybe get to how we can still use those kind of theories, where to justify.
Typical AI, that's not the case.
No, that's right. So there are other issues there. That's right. Yes, please.
I'm curious with the comment you said that, if I understood correctly, in flies it is symmetric, or in other...
Sorry?
If I understood correctly, you said it in flies or in other...
Yeah.
Everything is symmetric?
Not everything, not everything. So first of all, neuroscience is a giant field, and I know very, very tiny amount. And I learned about the fly brain only 3 months ago. I didn't know this weird thing was there in the fly brain. So it turns out in the fly brain, there's this central complex, which is supposed to have RNN realization, everybody believes. And when you study the neuron carefully, these neurons, the axons and dendrites are right intermixed. So input and output are right next to each other. They are not separated, from what I know. But this, my knowledge is of 2 weeks ago, so maybe what I'm saying is wrong. So take it with a grain of salt. This is as much as I know. Yes, please.
Yeah, so I imagine you were going into that anyhow, but I was just wondering, so what are the behavioral consequences of that? So why does that matter?
Don't know. I'm not Kepler. I would love to know. I would love to tell you why it's the case, but maybe, I'm going to overpromise massively. So maybe after a few slides, I'll try to show one toy example where we solve the advantage of this kind of a thing. Maybe we'll go there. And turns out we just published a paper on it. I can give you a reference shamelessly. It's a paper in Nature Communications that this weird connecting, this goes only one way, actually gives rise to a new kind of memory, which is highly energy efficient and very reprogrammable on an instant, unlike this kind of memory. So the memory is stored right here. According to our current understanding, the memory in the brain is stored in the synaptic strength. So how many vesicles will it dump? The probability of release, the quantal size, all of that changes. That's how synaptic strengths are storing the memory. That's very expensive because all these vesicles have to be recycled and all that stuff. So we found a different way, which is just an emergent property. The memory changes very fast, but if we get that far, I will talk about it. Otherwise, just take a look, called persistent activity and inactivity, some theories, blah, blah, something. It's on our lab's webpage, or ask me later. Okay? Any other questions? Yes, please.
So another, you seem to suggest that in most artificial models, that are symmetric, but
No, so
that's completely, I think, not the case. They can,
that's right.
Networks in particular are not something
that's right.
anybody ever implemented for any purpose.
That's right. So what I was talking about was Hopfield model or equivalent. Yeah. In those ones, there are strictly symmetric connection, and then the missing connections are so-called noise. There are deep networks I know nothing about. They do something else altogether. Now, the other important thing to keep in mind is that most of the deep networks that I'm aware of, and there are so many I can't keep track of it, is the activity of the node or the neuron that contains the memory. That's what is reprogrammed when you do backpropagation. But in the brain, the neurons' activity levels and so on, they are not really that modified. It's the synapses where the memories are. So the weights themselves are trained, but not the ReLU function itself. So that's a major difference between the brain and neurons. All right? So let me see. Now, so what happens now is that this releases neurotransmitter. There is equally interesting and complex stuff on the other side. The gap between this is universal. It's 20 nanometers. So everything, all neurons are separated from each other by 20 nanometers. That's a magic number, another nice magic number. So when the neurotransmitter is released, it has to travel the giant distance of 20 nanometers. And all kinds of funky stuff happens there in that 1 millisecond. So 1 millisecond, the action potential time scale, the synaptic release and reaching the postsynaptic target. Another millisecond, the neurotransmitters get injected, 1 or 2 or 10 milliseconds, and the time from which it's injected reaches the cell body, and then tens of synapses have to come together. One synapse cannot activate the output neuron, so you need at least 10 or 20 synapses typically to be activated at the same time for this neuron to cross threshold and fire its own spike. So that's where my idea of clock speed of a millisecond comes from, because all of this is on the scale of 1 or 5 milliseconds. Yes, please.
So what's stopping people from just building an artificial brain if there aren't that many neurons and there's not that much energy involved? Is it just
people are doing
architecture?
People are doing it actually. Several people have done that. So one guy at IBM has built that. There are people who are building neuromorphic chips. But there are issues. So where are the issues? So there are two places where there are issues. One place where there is an issue is that you want this programmable. And you want 40,000 inputs on a neuron. So when you want to have each neuron, you can integrate some linear, integrate in FIRE or CDVDT, linear differential equation on that. That's pretty easy. There are neuromorphic chips there, but to make these 40,000 synapses on a neuron and then make them programmable is hard. So Intel came up with a Loihi chip last year, which put in a biophysical learning rule called STDP, or it's kind of Hebbian, or whatever people know. So that's available, but that can do a few of them programmable. So it's the input-output is the problem. How do you take all that out? Yes, please, at the back.
Just a comment to Richard's question. So it's not just this. There is different layers. There's cytoarchitectonic differences in different parts of the brain. There are neurotransmitters. There's so many chemical activities. It's not simple.
So that's right. So right now I'm talking from a very simplistic view of the brain that's closest to AI. Now, as Ida pointed out, as you start going deeper in the brain, you find all kinds of other weird things. But I'm talking about the lowest level. Yeah, even that is hard. Yes, please.
Maybe one more comment. Even if you could somehow do this, right? The brain comes with some predetermined structure.
Yeah, that's right. That relates to that. That's right.
Not even on the level of neurotransmitters.
That's right.
What connects to what? Different layers, different levels.
Right. That's right. And at the same time, all of you must have heard of it. There was this English patient. We had this neocortex, is all these 7 layers. I call it 7 layers of sin. There's 7-layer structure, but we don't know why we need 7. Why not 3? Why not 2? So there was this English patient, and he went for routine checkup, and he found his whole skull brain was like about that thin. That's it. And he was perfectly normal Englishman. I mean, that says something about Englishmen, but I didn't say that. It's 200 years of slavery, here I come. so there are amazing level of things going on, and we don't know how much of that's needed. It's not a small perturbation that the whole skull, and there's another paper that just came out recently, another guy, this time American, and I had this skull, the whole brain shrunk to like 3 millimeters, the whole thing. Seems totally fine. Yes, please.
So in general, like when you look at these like biological artifacts or facets, how do people really know what aspects are kind of the brain works despite them, or the brain work because of them?
Another good question. See, that's why all of you should work in this. You can ask amazing questions and then spend 4 years solving it. So we don't really know. There is a lot of experiments. People have done amazing amount of stuff. This thing I'm talking about is 100 years old. More recent things, people can come in through fancy technology. We and many people can do that. I can make either one synapse active in an awake animal out of God knows some billion, or one neuron active and shut down and so on. So there's a lot. But at the end, if you were to ask me to be Kepler, I'm at a loss. There's no Kepler here. So it just, yeah, this works and that also works. But at the same time, it's not entirely hopeless. So if I get there, I'll show you some examples which are very robust, which can do lots of cool stuff. So including predictive coding, which is one of the central stuff. So we'll come to that. Yes, please.
What is the simplest organism that has this?
The simplest that I can note is called C. elegans, which is heavily studied. Now, the cool part about C. elegans, it's a worm, makes a lot of people sick. There are many varieties. Its brain is only 302 neurons, so very small number. But the funny thing is that in the C. elegans brain, it has all this stuff, but the neurons don't generate spikes. They just graded potentials. So that is a later evolution, and that's going on in your brain too, actually, by the way. So right there in your eyes, you have a 6-layer network in your eyes. The retina is this 5-6-layer network already. So rods and cones get input. Rods and cones take photoreceptors, translate light into some signal, but the signal rods and cones send to the rest of the brain is analog. The next layer is also analog. It's the 3rd layer that starts to generate spikes. So people think the reason you want to generate spikes is because you want to send signal over long distance, and if it's AC, via Edison versus Tesla, AC is easier to send, blah, blah. So, yeah.
What do we know about how the synapses change with experience?
Yes, a lot is known, and I'm going to get into that. Any other questions?
Yes.
Yes.
A clarifying question. So at the synapses, the cells do not satisfy action-reaction principles.
That's right.
Okay.
That's right.
And the reason why I ask is because, kind of like Richard, I've always wanted to see this as a directed graph, and there's non-local vector calculus.
That's exactly right.
Dynamics on that.
That's right.
That has action-reaction principle abilities.
I know, because we built all our mathematics based on the world we experience, which obeys Newton's first law. But our brain decided to just violate massively. All right? Any other things? So then there is one more level of biological fact that you want to know, that there are actually, roughly speaking, 2 kinds of neurons or nodes. In AI, all nodes are more or less equal. There are 2 types of neurons. This is called the Dale's hypothesis, D-A-L-E. If you want, you can Google it, which says the following, that if there is a neuron, and there are neurons, and each neuron makes roughly 10,000 synapses, either all of those 10,000 synapses are such that put positive ions in the other neuron, so its effect is to activate the other neuron, make it more depolarized, more positive, so it is prone to spike, or all 10,000 synapses are injecting negative ions. And I cheated there, it's not necessarily positive or negative, but I want to keep it simple, so we're going to forget potassium and all that weird thing. So there are 2 types of nodes in the brain. One type of node, both of them generate spikes, but one type of node has a specific kind of chemical and a specific kind of receptor, so that as a result of it, that synapse, whenever this is activated, it always injects positive ions, makes the other guy more active. The other kind injects such that it makes it less active, overwhelmingly true across the brain. All right? And then one final fact that you may want to know, I want to keep track of time, I was going to tell you a lot of things, but so the other thing is that evolution has found one more trick, which is to package this group of this, and by the way, neurons that inject positive ions, I call excitatory, because then to excite the other neuron. Ones that inject negative ions, I call inhibitory, they are called inhibitory because they inhibit. The ratio of roughly excitatory and inhibitory neuron is roughly 10:1, more or less. There are 10 times as many excitatory neurons as inhibitory neurons. Now, why the hell would you put inhibitory neurons in the brain? So standard idea people had is to say, wait, if there are only excitatory neurons, they can have a recurrent loop, they can excite each other, and the brain can fry itself. But that idea is actually going to the roof as we understand more and more the dynamics of circuits. Inhibitory neurons actually are more likely to make brain go epileptic, to fry itself in a complicated way, and that's got to do with dynamical systems. So I won't have time to get into that. These are playing some crucial role in actually neural coding. And if you now go further and if you look inside the brain and say, all right, let's say I'm looking at this, the earliest studies was this thing is moving and so on. If the neurons are responding to this, it's the excitatory neuron that seem to encode information about whatever is happening there. Different smells, different colors, different light, different objects. A neuron, excitatory neuron will fire differently based on different colors, shapes, smells, and so on. Inhibitory neurons more or less seem to not care, broadly speaking. So there's something funny going on in the brain as well, which is not in machines, which is that there are two types of nodes. Yes, please.
Is the distribution uniform to the brain, or
no? In the neocortex, which is what most AI people restrict themselves to, it is more or less uniform. But if you go slightly deeper than that, then there are specific brain areas which are purely inhibitory. And there are others, but let's say for practical purposes, this is 80-20 or 90-10 is pretty much accurate, by and large. Yes, please.
So maybe I'm missing something, but doesn't that just feel like essentially you're enabling positive and negative contributions from an output of if you model some output of a neuron as a tangent function, you need positive and negative weights.
But that's the thing, that's right. If you model as tangent hyperbolic, that's exactly right.
Then I guess you're saying that every neuron either contributes negatively to every one of its.
That's right.
Well, that might be just a kind of aspect of biology, right? It can't both inject positive and negative ions.
No, it's not that. These are specific. It has gone out of the way to create a very complicated machinery. This thing injects a chemical called glutamate. Its receptors are very specific proteins called AMPA and an MDA, metabotropic and all that stuff. For inhibitory synapses, different injects another kind of acid. These are all acids called GABA. It is a distinct kind of receptors. This hole will only let chloride in. For inhibitory synapses, for excitatory synapses, it only let sodium in and calcium every now and then. So it's not an accident. And let me give you the short version of the story and move on. The whole advantage and disadvantage here of all this keeping is that neurons are operating with impulses. They're not smooth functions. Their computation are based on impulses. Pop, one impulse and so on, and that's where inhibition shines quite a bit. So it won't make any sense, but let me move on if it's okay. Somebody had a question.
So by node, you just mean neurons?
The word people use called nodes. Yes, I mean neurons. In the brain, that will be the equivalent.
Okay.
People call nodes.
The edges is neurons.
Yeah. That's just I'm trying to build a dictionary of what people might be familiar with. Okay. So this group of excitatory and inhibitory neurons, about 1,000 of them, or 10,000, form a column. And then what evolution is, and within those 10,000 neurons, there are 7 different layers, and they have different connectivity pattern. And what that's like, as I was telling somebody this morning, that's like the op-amp or a little chip. Then what the brain does is, if this is about less than half a millimeter by 1 millimeter, it just keeps building more and more and more of those. So that's the organizing principle of the brain, to have about 10,000 of these in some column, and then just put lots of them, and then whether you go to visual cortex, auditory cortex, and so on, it's the same. Yes, please.
What do you mean by column?
So these are all the neurons. They're literally
Literally line up.
they are like vertical here, and there's strong connection within it. There's a specific architecture here, and then that column it repeats everywhere. So if the brain looks like a sheet, which if I can take my neocortex unfolded looks like a sheet, is 1 millimeter thick, and God knows tens of centimeters square.
Physically, they literally line up like a column.
Yeah, exactly. That's right. It's exactly the same column that repeats everywhere. Whether you go to your auditory cortex or visual cortex, you are hearing things or smelling things or anything, it's doing the same thing. All right? So let me give you some. Now, there is one more thing that the brain does which machines don't do. So this is something I got interested in quite a bit. So this is an actual neuron now in the brain, right there. From that part of the brain right there, it has a name called parietal cortex. It does complicated visuospatial association, fancy stuff, and so on. And this is the scale bar here. This 250 micrometer. That's a neuron right there. That's the surface of the skull. That's the cell body. These are all the dendrites, and that's very axons are much thinner. You can't see them. That's the axon. And all this varicosity that you're seeing here is not bad microscopy. Those are the tiny little synapses which are 1 micrometer that are sticking out like that. So you put a pipette here and you measure the signal coming out of one neuron. This is time in seconds, and the animal is anesthetized. It's doing nothing. There's no stimulus, the room is dark, nothing is going on. Animal is just lying there, and in red is the membrane potential of that neuron, so membrane voltage. Here is membrane voltage in millivolts, and here it is in some number of standard deviations. So we don't have to worry about units. So z-score membrane voltage goes about 3 standard deviation, fluctuation, boom, it goes up, then comes quiet again, then goes here and boom, and so on. So brain, when nothing is going on, not only it is active, it's active in a very periodic way. These are called up and down state oscillations. The brain behaves like a 2-state system. And this happens to you when you go to sleep as well. So when you go to sleep, for some reason we have a need to sleep. Our brain is still active, but when we are sleeping, majority of our sleep is called non-REM sleep. That's the majority is 80 to 90% of sleep is where we don't dream. It's only about 10% or so of the sleep where we dream. Babies dream a lot. As we grow older, we dream less and less. So you're literally having less and less dreams. Oh, it's a biological fact. Apt enough. And then, so this is called the non-dreaming sleep or non-REM sleep. REM sleep is where you move your eyes and so on, another story. So what happens is that the brain behaves like a 2-state system. It becomes very active, and it becomes so active that every now and then, about once in every few seconds, it generates an action potential, otherwise it does this. Now what we did is, when I say we, is my wonderful collaborator, Thomas Hahn, is that we put another electrode far away from this neuron. Like this neuron is here, the other electrode is like, God knows, 2 centimeters away, or 1 centimeter away. This is the average activity called the EG, or the local field potential in blue. You can see that both of them are doing the same thing. So what this shows is that the most common thing your brain does, the most common thing is this. As soon as you close your eyes, you stop moving around, instantly your brain will do that. You don't even have to go to sleep. Your brain likes to behave like the brain by I mean neocortex only, sorry. That's a bad mistake. This is only the neocortex. The rest of the brain does other complicated stuff. And this is so prevalent that even when you are sitting here and listening to me, and if you simply were to calm down a little bit and not move your body or something and not blink too much, instantly your brain will want to do this. So there is something fascinating about it, that the neocortex, which is a big chunk of the brain, wants to behave like a 2-state system, boom, boom, and this period is more or less 2 seconds. The exact structure changes. So this now we have a handle on the ground state of the brain, right? Ground state of the brain, ground state of the neocortex, ground state of the neocortex, when there is no stimulus, there is no behavior, is to behave like a 2-state system. Yes, please.
How do the cells synchronize? I mean, they're doing it at the same time.
Yeah.
And they're far away.
If we get time, I'll show you a simple theory, because if I want to be Kepler, I still have dreams of becoming Kepler, so. We have tried to make a simple theory, and it turns out one can make a really powerful simple theory of how this does. And let me give you the answer right away, because I may never reach the answer because there are too many promises. So if you simply take one excitatory neuron and one inhibitory neuron, and you say, I club all the excitatory neurons in the brain together as one big fat excitatory neuron. Other all inhibitory neurons together, one fat inhibitory neuron. The average activity of all the excitatory neurons, so many spikes across all of them, I can assume that it's a continuous function. Same for all inhibitory neurons. If you simply take all the excitatory neuron as a continuous function, maybe some sigmoid like it and nothing else, and all inhibitory neuron, that will just oscillate very, very fast, which is a bad thing. Because now the whole thing goes push, pull, and so on. This excitatory neuron will excite the inhibitory neuron, that will shut down the excitatory neuron, so it will shut down, which means shuts down the inhibition, which excites the excitatory neuron, so you'll have very fast oscillation, which is bad. But the fact is, another well-known fact is that there is a nice separation between the functions of excitatory and inhibitory neuron, which goes back to your question, that all the excitatory neurons have specific currents in the cell bodies, not in the synapses, but in the cell bodies. So if you just took an excitatory neuron, put it in a slice, took out all connections, and injected current for just 1 second, and long enough current, so large enough current is going to fire spike, all the excitatory neurons will start firing spike like gangbusters right in the beginning, and then they will stop firing. And by the time you put current for like 5 seconds or something, they'll stop firing altogether. They get fatigued. The mechanism that generates action potential is complicated. It doesn't work anymore. But that is true only for excitatory neurons. The inhibitory neurons, if we did the same experiment, injected current for 1 second, they don't get fatigued at all. They keep going more or less at the same pace. And if you simply put in that much fact, that you take one average excitatory neuron, one average inhibitory neuron, and only the average excitatory neuron gets fatigued, you can get these slow oscillations. These are called slow oscillations, delta oscillations, non-rep sleep, it has many, many names. I'll try to show you how we can calculate and how that can be used to create new kind of memory as well, which is highly energy efficient. Maybe we'll get there. Yes, please.
Yeah, I was curious if it's synchronized with your breathing. Like breathing's about
there is, absolutely.
Like in meditation.
That's right. So yeah, I have experimented with that myself, and you can do that exercise too. There are machines available for $200. You can put it, this EEG called delta band is so synchronous and large, you can pick it up with an average cheap thing. And now what you can do is, you can take the Hilbert transform of that, so it gives you magnitude of the signal. And now we can become true Americans. So this toy allows you to compete with your friend in who has better delta oscillation. So you can have a little knob, that knob goes one way if I start to have better delta oscillation. So it's gamified, the best way to learn. Yeah, sorry.
This is to the person was asking about inhibitory neurons earlier. The earliest evolutionary evidence of that is in coral polyps who don't have brains, but they do have excitatory and inhibitory neurons, and that's used for closing and opening the same mouth.
That's right.
They look like a star, and they have a mouth in the middle. If they perceive food outside, they open mouth, and that's excitatory neurons. The same muscles need to close when they want to close the mouth, so the inhibitory neurons do the same thing, but the reverse. And it closes when it perceives food in stomach.
That's right.
So it's like the simplest neural circuit evolutionary that we know
That's right. People have looked at, that's right. That's right. And in fact, it turns out that this whole excitatory inhibitory separation is not valid in our brain too when we are born. When we are born, glutamate is actually, and GABA, the separation is no longer there. So that leads to another thing. As we develop, we start to evolve. There's a whole field of neuroscience called evo-devo. To say if you want to look at evolution of things, just look at the development of a single individual from fetus to head, and many of the similar phenomena are seen there, right? So that's another thing. So now it turns out that the brain generates these oscillations when you are not doing anything, but that's not the only time. If I were to put an electrode in the brain, as we do routinely, right behind you actually, it's so funny, I only realized now, that building, if you simply were to go to 5th floor right now, that's where these experiments are going on. These data are recorded right there. So not this one, but similar. So turns out that if you were to now, this is the whole brain's activity level become more or less two-state only when you are not doing anything. As soon as you start to do anything, the brain develops a wide range of rhythms, polyrhythms. EEG looks like 1 over F. So the voltage versus frequency plot looks like 1 over F, raised to some beta. So another set of interesting things. So let me conclude this part and maybe show you a couple more interesting things about what's going on. So maybe I don't show you this. How do neurons then encode information? I want to quickly go through that. So actually, before that, I should show you how individual neuron responds. Sorry about that, I have arranged. So here is that one experiment somebody did. So the brain is actually wiring up, as I showed you an example right there, but the wiring up is started up even when you are developing. So this is another thing that's non-trivial. This experiment was done by Colin Blakemore. It was challenged later on by a lot of people, but the idea is more or less the same. So what's the experiment? So you have a kitten, and it is wearing this thing, so it can lie down on the side and so on. And the kitten has a brother, male kitten. This was all allowed 60 years ago. Now this is harder to do. They are kitten and so on, animal rights. But same thing works for us. Nothing nasty is going to happen, so breathe easy. And all that happens is that this kitten grew up in a little cylinder with only vertical stripes. And the kitten's brother grew up in a cylinder with only horizontal stripes like this. So for first 3 months, they saw the world perfectly. And this kitten, except it saw mostly vertical, it still tries to lie down, which messes up the experiment a little bit, but whatever. And then at the end of this roughly 3 months, they swap the kittens. And turns out that the kitten, once they go to horizontal orientation or the horizontal kitten goes to vertical, they don't see the wall. Just head in there. So turns out that simple facts, this is vertical, this is horizontal, just binary, we don't learn it. We are not born with it. We have to learn it. And that learning happens rather quickly through some wiring in the brain, through something called synaptic plasticity. And that happens within the first 3 months in the visual cortex. 3 months in kitten, maybe a couple of years in humans because things slow down. So this thing, synaptic plasticity, that rewires the brain, which rewired your brain just now when I started this talk, where we made sure that you see very different things and so on. That rewiring occurs through change in synaptic strength.
Sorry, you said that they don't see a vertical, they don't have a vertical.
Yeah, the one that didn't experience, they don't see it. And turns out this synaptic plasticity is different, it's regulated. So in the brain, you have a deep network. This experiment where this orientation is perceived, Nobel Prize winning works from Hubel and Wiesel showed that this orientation, vertical or horizontal, is perceived in the very first part of a visual cortex called the primary visual cortex here. That's where it is perceived. Hang on one second. What happens is that circuit needs to be laid down within the first 3 months or so of life because there is a ton of synaptic plasticity. But once this is laid down for the first 3 months, then visual cortex synapses don't change. And there is a plasticity gradient. So if you go to the earliest stage of the brain, such as primary visual cortex, so first or second layer in a deep network, then those learning gates shut down early. But as you go further and further in the developmental stages, all the way down to prefrontal cortex or hippocampus, those remain plastic for much longer and longer, which says something about how things are learned. You had a question there. Yeah, okay. So, and that's why, so then let me actually get rid of this and show you one little thing, and then we should take a break. How much time do I actually have? Sorry, 3 minutes. Okay, so let me just explain. So then what happens is, here is a classic experiment that everybody should know. This is an experiment done by Lord Rayleigh, 1926, almost coming to 100 years. So he took a frog, and now instead of sensory cortex, motor, he took the frog's leg, put an electrode there. The synapses are not only in your brain, but also in your body. The brain generates signal, form synapses on your muscle. When the neurons are activated, the muscles contract and so on. So the same idea happens here. Those action potentials are coming all the way here. And when your muscles move, those signals are carried to the brain also by action potential. So if you want to really study action potential, you don't have to put electrode in the brain. Take a tiny, tiny wire and inject into this nerve, and you can actually study how your arm movements are encoded by neurons which are going all the way to the brain, 1.5 meters. So what he did was took the frog leg and put some weight. So he put 50 grams, and on an average, when he put 50 grams of load on the leg, it generated 1 spike every now and then. And then if he put 100 grams, it generated 3 spikes. When he put 250 grams, it generated 4 spikes. And when he put 500 grams of weight on the frog leg, it generated more or less 5. It's some funky pattern. It was highly variable. So he came up with a very fundamental rule, which is this. So x-axis is seconds. Y-axis is how often this neuron generated a spike. And notice this is not generating at the rate of 1 spike per second. This is going all the way to about 20 spikes per second. In this case, it is a small amount of weight that was put in for about, notice, from 5 seconds to 20 seconds. And what you see is something fascinating. As soon as you put the weight on, the neuron cranks up its firing rate. It takes quite some time for it to crank up to the maximal value. And then the neuron starts to get fatigued. And there's a lot of stochasticity here. It doesn't generate, even with a simple experiment, it's not visual cortex and this and that, it's just goddamn weight and just this muscle. There's a lot of stochasticity here. There's a lot of variability. Even in this extremely simple experiment, there's some systematic change taking place over time, and there is noise taking place over time. So as a result, the fundamental belief in neuroscience is that neurons, no matter where you go, all the way down to your muscle, they encode information using something called the rate code. The rate code is that for some weight, small amount of weight, low intensity, horizontal orientation, perfume of a rose versus vanilla, different neurons encode all this information using a rate code. So for maybe a neuron in the olfactory bulb or piriform cortex or orientation thing, will fire at the rate of, on an average, 1 spike a second for this weight or for this color or for this smell, and it'll fire at a slightly different rate for a different color and different fragrance and so on. Highly stochastically. How stochastic is it? People measured the standard deviation. Many, many times you present the smell of roses, so many, many times you present the vertical orientation or the horizontal orientation. For just the vertical orientation, you present it 100 times. You measure the mean firing rate. You measure the standard deviation. Divide the standard deviation by the mean. It gives you a dimensionless number regardless of neuron firing rate. That number is unity. This is called the coefficient of variation. Right? Standard deviation divided by the mean. Because the numbers are all positive, so you can do that. All neurons, by and large, are highly variable. Whether the neuron fires on an average 1 spike per second or 50 spikes per second, on an average, the neurons vary their firing rate for the same boring stimulus, such as the weight or this oriented bar, by as much as its mean value. Fundamental fact, standard deviation is more or less equal to the mean, even though the firing rate or the spiking probability changes by at least an order of magnitude. So these are some why the brain wires it up such a way, interesting questions. So let's just stop here and continue next time. Yeah.